Hello, everyone. My name is Josh Sommer. I'm co-founder and executive director of the Chordoma Foundation. I'm really excited to join you today to talk a little bit about the Chordoma Foundation's research, where we've come and where we're going. But first, a little bit of background about me. I am joining now from Durham, North Carolina. Here you can see a picture of our city and the arrows pointing to the Chordoma Foundation's headquarters and lab. I live here now with my wife and two children, which is something that I never would have thought possible 18 years ago when I was diagnosed with a skull-based chordoma at the age of 18. I was very fortunate to have a great surgery and a complete resection. But after my diagnosis, as I learned more about the disease, I became somewhat shocked and disillusioned by the statistics that I learned about a high rate of recurrence, 8-year average survival, few effective therapies beyond surgery and radiation. And at that point, I became determined to try to do everything in my power to try to change those odds. It just so happens that at the time, the only researcher in the United States that had a federal grant to study chordoma was, of all places, at Duke, right where I was studying as an undergrad. So this provided a great opportunity. It was incredibly serendipitous. I ended up joining this lab of Dr. Michael Kelly. It was an incredibly eye-opening experience to see what was possible in cancer research at the time. But also it was eye-opening in that it really highlighted the challenges that researchers interested in chordoma faced. We didn't have access to adequate materials like tumor tissues, cell lines, animal models. We were working in isolation. Obviously, funding was limited. And an interesting a coincidence actually was that the only cell line we had in the lab of all places came from the University of Ulm, where Dr. Moeller and Dr. Brudeline and others on their team had succeeded in creating UCH1, the very first chordoma cell line. But quickly it became obvious that there was a lot to do, and only so much that one lab could achieve on its own, that there was a need for many more labs and researchers and companies to bring their expertise to bear on this disease, that it would require significant resources, it would require organizing fellow patients and family members, et cetera. And so while working in the lab, I connected with a number of other patients and family members through an online support group. Several of us met in the fall of 2006 at the National Organization for Rare Disorders annual meeting. There we were inspired by one another and inspired by the work that had been done by other patient organizations. Several of us got together and in 2007 started the Chordoma Foundation. And from there, this movement has really snowballed, now involving thousands of patients and family members worldwide, hundreds of doctors and clinicians, thousands of donors, volunteers, et cetera. And at the Chordoma Foundation, we have a very clear mission, which is to improve the lives of those affected by chordoma and lead the search for a cure. And we do that through three interlocking and interdependent areas of focus: patient services to create a better experience for patients, healthcare improvement to enable healthcare providers to deliver better care for patients, and research ultimately aimed at developing better treatments and a cure, all with the goal of enabling patients to overcome the disease and maintain their quality of life. Very briefly, to touch on our patient services, we offer a comprehensive suite of resources and services spanning from educational materials through support groups to our patient navigation service, and more recently, financial assistance to participate in clinical trials. Additionally, we've worked with leading clinicians across the world to develop consensus guidelines. The first ones were published in 2015 by a group of over 60 international treating physicians. There's an effort getting underway now that we're leading in collaboration with the Istituto dei Tumori in Milan to update these guidelines after 10 years to reflect the latest understanding of the disease and recent research findings. And then our research efforts are really centered around developing better drug therapies for chordoma. We know that despite the high rate of recurrence, many patients with chordoma, surgery and radiation can be quite effective and have really become quite advanced. What's missing is effective systemic therapies, effective drug therapies that can help either control recurrent disease once it's no longer amenable to being treated with surgery and radiation. Eventually, perhaps to augment surgery and radiation to help prevent or reduce the likelihood of recurrence. And eventually, in the long run, perhaps serve as a safer and more effective alternative even to surgery and radiation. We know from our understanding of cancer in general that it's very unlikely that a single therapy is going to solve the problem. We know from other cancers that more than likely a combination of therapies are going to be required, that we're going to need multiple therapies to be personalized for individual patients based upon the makeup of their tumor. And perhaps we're going to need to treat patients sequentially so that if eventually the tumor becomes resistant to one therapy, other therapies are available that could be effective. Now, if you zoom out and take a big picture approach, there's several different paths or finding better therapies for chordoma. There's about 3,000 drugs that are already on the market that could be used off-label in many countries for chordoma. There's a larger group of about 7,000 drugs that are being developed at any given time, about 1,300 of those for cancer. And then, depending upon the biology of the disease, it may turn out that new drugs need to be developed, and there is a plethora of very powerful new technologies and therapeutic modalities that are emerging that are providing new opportunities and new ways to attack cancers like chordoma. Now, which of these paths we need to go down is ultimately going to be dependent upon what we learn about the biology of the disease. But what we can know is that regardless of the path, there's going to be several key areas of research where progress is going to need to be made. And in order for patients to benefit, it's not enough to fund or support or advance research in one of these areas. Seamless progress is needed across this entire research and therapeutic development continuum. So to enable that, there are a lot of things that are necessary but not sufficient. Conventionally, patient organizations, foundations have supported research by raising money and providing funding. And that is extremely important. However, that's only a part of what's needed. It's only one of the key essential ingredients. In addition to research funding, it's absolutely critical to facilitate knowledge exchange and collaboration among the research community to recruit new researchers into the field. In addition to that, there are a core set of enabling scientific resources that are needed, things like tumor tissues, cell lines, and animal models, which I alluded to earlier. And we've created centralized repositories of all of these resources and made them available to over 120 researchers worldwide. In addition to that, more recently, we've developed a number of research platforms. These are shared research capabilities that prevent researchers who are new to chordoma, for example, or companies who maybe have a relevant drug or technology from having to recreate chordoma-specific capabilities themselves. Instead, we've created centralized capabilities that could be accessed by researchers worldwide. And in doing so, we lower the barrier to entry for new researchers and companies and make research much more efficient and faster. And then finally, there are a number of things that the foundation does to facilitate patient participation in research, which is ultimately critical for in making clinical trials successful, for learning about how the disease progresses, for contributing to the development of new cell lines and animal models, et cetera. With this collection of resources and ways in which we're able to drive research, we are collaborating with researchers across the world by providing funding, by providing enabling resources, et cetera. So here you can see a snapshot of the researchers that we've supported or collaborated with across a number of different continents. And in particular, in Europe and in Germany, there are a number of very promising research projects going on in Heidelberg, in Ulm, in Freiburg, et cetera. In addition to the work that we support among academic labs and companies, as I mentioned, we've recently set up our own lab, Chordoma Foundation Labs, here in Durham, North Carolina. This is the only lab in the world that's 100% focused on chordoma research. And the reason we set this lab up is because for researchers with cutting edge expertise, capabilities, et cetera, to be able to apply those capabilities to chordoma. But in order to do so, they oftentimes need to create chordoma specific capabilities in their own lab or become develop expertise in chordoma. That is a time-consuming and costly undertaking. Instead, by having our own lab with a set of capabilities and deep expertise specific to chordoma, we're able to offer those capabilities to researchers and companies who are maybe new to chordoma or maybe who are never going to become focused on chordoma, but nonetheless who have something important to contribute to this field. By centralizing these capabilities, we're able to benefit from economies of scale and increase the efficiency of the experiments that are performed. And in addition to that, when important research is neglected for various reasons, because it's insufficiently incentivized within academia or industry, we're able to pursue that on our own urgent timeline. So with the combination of initiatives that I've described, in collaboration with this now energized and very capable field of over 300 chordoma researchers across the globe, we've come a long way together in the last 17 or 18 years. We've gone from having basically no research enabling resources, except for that one chordoma cell line from Ulm that I mentioned, to now having abundant resources, including 26 cell lines, 13 chordoma mouse models, biospecimens, that's tumor tissue, et cetera, from over 200 chordoma patients. We've gone from knowing almost nothing about the biology of the disease to now having identified a plethora of targets that point to opportunities to apply existing drugs or maybe drugs that are in clinical development, as well as the identification of an Achilles heel for chordoma, a target that chordoma relies on absolutely, but is not present in normal tissue. This is called brachyury, and it represents perhaps the greatest opportunity for effective and potentially curative therapies for chordoma. A number of years ago, there was no research going on developing drugs for brachyury, and it was assumed that brachyury was an undruggable target. And now, thanks to the research capabilities that have been developed, research that we've supported, as well as the work going on in Chordoma Foundation labs, there are now multiple brachyury drug discovery programs, and the very first compounds have been identified that actually can interact with the brachyury protein and potentially serve as templates for drugs targeting brachyury. We've gone from having no drugs tested preclinically, that is in mice or cells, to now having tested more than 90 different drugs preclinically, 10 of which have demonstrated strong preclinical activity and have advanced into clinical development. When the foundation started, there was one clinical trial that had ever been done. And now over the last 15 years or so, we've helped initiate a dozen clinical trials with 5 more that are being planned. Encouragingly, this culminated in, well, whereas in the past, systemic therapies were ineffective for chordoma, now in the last several years, 3 therapies have been identified, which for a subset of patients demonstrate clinical benefit. Here you can see this group of 3 drugs highlighted on the right of the screen, each of which for between 10 and 15% of patients induce significant tumor reductions. But as important, reducing the tumor volume in some cases can allow patients to live for an extended period of time without tumor progression. And this is not abstract. These are not just abstract numbers. These are real people who are part of our community. Here you can see 3 individuals who have benefited from these drugs, each of them for 3 years or more, and in some cases, many more years than that. So, made progress, but there is still a long way to go. Obviously, we need drugs that are going to work for more patients, that are going to be more beneficial, and that are going to induce or provide benefit for a longer period of time. Now, thankfully, because of the research that's being done, there's a growing pipeline of promising therapies that are emerging from the lab into the clinic. And looking forward, we're moving from a regime where the drugs that are being tested are largely empiric, basically investigators having an idea, testing it in the clinic, to now having clinical trials based upon very strong preclinical or molecular rationale. We're going from a regime where we expect a one-size-fits-all approach, one drug to work for all patients, to recognizing that we're going to need to find biomarkers or ways of predicting which drugs work for which patients so that we can personalize care. Again, going from this concept of a single agent to combinations, and increasingly shifting from repurposing old drugs to now exploring drugs that are either in clinical development or actually developing new drugs that could be targeted specifically to chordoma. And because of what's happened over the last years or so, we've really reached a number of turning points that enable us going forward to realize opportunities that were completely out of reach even just several years ago. Looking ahead over the next 3 to 5 years, we have a set of audacious goals now to complete the remaining research enabling infrastructure needed to take therapies all the way to regulatory approval, actually leading to the first drugs approved to treat chordoma. In the next 3 to 5 years, we're going to position chordoma patients to benefit from extraordinarily promising advances in technologies like artificial intelligence, as well as new therapeutic modalities, things like antibody drug conjugates, radio ligand therapies, et cetera. Within that time frame, we expect to be able to bring, with adequate resources, the first brachyury drugs into clinical trials. And again, with adequate resources, we are on track to be able to bring 6 to 8 new therapies into clinical trials within that time frame. And then in parallel, on research that's going on in CF labs and elsewhere, we are on the cusp of identifying ways to be able to match patients with the therapies that work for them. And I want to highlight really extraordinary work that we've done with the University of Heidelberg, DKFZ, and the Technical University of Munich. In fact, leading to a publication recently led by the Technical University of Munich that was, I should say, a manuscript that was just recently submitted, identifying a way of predicting response to a class of therapies called EGFR inhibitors, drugs like afatinib and cetuximab, et cetera. So this is a very promising area of emerging research as well. I think tie this all together, and what does this mean for those of us affected by this disease? I think now there is more reason to be hopeful than ever before. And really that is thanks to the incredible community that has rallied around this mission. Together, though we are small, we have the ability to improve the outlook for this disease. And I'm so thankful to the thousands of patients and family members across the world who contribute their time, their talents, their resources, whether it be donating, whether it be doing fundraisers, et cetera. Every single one of us has a role to play, and every single one of us can make a difference. So thank you so much for making our work possible. I'm deeply appreciative. If there's anything we can do to support you, whether it be through your journey with chordoma, your involvement with fundraising or philanthropy, or simply if you have questions for us, please don't hesitate to get in touch or to follow us online. I hope that you have a great time today, and I look forward to being in touch. Thank you so much.